All right, let's go ahead and get started. Left off with the Echincococcus, right? Talked about the life cycle. Canines, definitive host. Intermediate host it's their food source. So for Echincococcus granulosus, typically find them in large herbivorous mammals. For Multilocularis, tends to be in rodents because Multilocularis primary hosts are foxes. Felines are secondary hosts. All right, and on Monday I said that this was probably an adaptation that increased transmission because you have a host that's being consumed by a pack animal. All right, and if you're trying to increase your transmission, why not have some sort of stage that reproduces asexually inside the host so that you can infect the entire pack? Added benefit is that as this asexual reproduction occurs, cyst is going to enlarge, which thus confers some sort of pathology, namely slowing down the animal makes them more susceptible to predation. So that larval stage is the hydatid cyst. All right, and the hydatid cyst we said it has this thick acellular layer, all right, resistant to host immune responses, and then just inside you have a germinal membrane, an inner membrane that ultimately gives rise to this entire structure, the brood capsule. Now, as the brood capsule pinches in and forms this bowl, you will have additional germination happening, giving rise to the protoscolices. Each one could potentially produce an adult. It's attached to a stalk. The stalks can break, and then you could have the collection of these brood capsules settled down in the cyst as hydatid sand. Now, difference between these two, between granulosus and multilocularis, comes down to their budding behavior. All right, so all of them exhibit this internal budding. They're all going to exhibit these brood capsules with the protoscolices. If they didn't, you wouldn't have your protoscolex develop, and you wouldn't have the transmission. But with granulosus, we have what's called a unilocular hydatid cyst, meaning we have internal budding only. It's going to grow, it's going to expand as kind of like just a large ball, you can think of, a fluid-filled bowl. All right, and as it continues budding, it's going to increase in size, continue increasing in size. Multilocularis is called a multilocular hydatid cyst, also called an alveolar hydatid cyst. All right, we have internal budding, but what this cyst does is instead of like enlarging in like a ball shape, it kind of spreads in more of a fluid, easy movement shape, which allows this to kind of reach into all the nooks and crevices of our internal organs. It fills in the spaces. And when it does that, then those small areas that kind of get in, if there's a pocket, it kind of spreads in there, and then it can form its own pocket that then pinches off and metastasizes and potentially spread to other parts of the body. So if you can imagine, a very small part pinches off, gets into the circulatory system, and then gets carried to the lungs. The unilocular cysts can't do that. They're kind of in place. Multilocular cysts could. All right? And the reason is that instead of having this thick acellular layer like we have in the unilocular cyst, I will. The multilocular cysts have actually a very thin outer wall, and that gives it its I don't want to say fluidity, but the ability to kind of move and find its way into these crevices. Now, in each of these pockets in the normal host, you're going to have those pockets developing a few protoscolices. All right. In the natural host, these small chambers are going to be fairly uniform in size. and they're going to be a lot more resistant to pinching off. In like humans and other non-natural hosts, now you have the possibility of these things pinching and metastasizing, and oftentimes those chambers that end up pinching off, they don't have any protoscolices, they don't ever develop protoscolices in those hosts. So you can imagine that these things, if they get into a host, it can cause pretty significant pathology, even in a human host. And that's going to be what we talk about next. I should say, most common site of these things is the liver, typically where they go, where they start developing. And you can imagine with the 3 lobes of the liver, the multilocular hydatid cysts can kind of fill their way into the various nooks and crannies, and also the structure of the liver. Who's had anatomy? Right? So if you remember, the liver has, we have models of the liver, and inside you've got your hilus. Is that right? Center of the liver? Yeah. No, part of the liver. Hilus is that the term? I'm trying to remember.
Hilum?
Oh, yes. Hilum. There it is. Hilum. If you can imagine one getting in there and then spreading into that structure, and man. But yeah, the liver tends to be the primary site of infection. With Echincococcus granulosus, the lungs is also a popular site. Right. You get this? All right, so humans can get infected. And if we get it, it's called echinococcosis or hydatid disease. So hopefully kind of recognize that hydatid disease is because this is being caused by the cyst themselves. Now, the infection itself can be latent for years. And what does that mean? It means it doesn't show any sort of outward pathology. We don't notice it. Right? It's hidden for years. And then all of a sudden it starts manifesting the pathological effects, or we start seeing it. Now, the overall effects just kind of depend on the location of those cysts, or where they are. But you have to imagine now is that these cysts, you have the asexual reproduction, the internal budding that's happening. They're growing in size, and no matter where they're at, you're going to have that enlargement of size and start applying pressure on the internal organs or wherever they're located. So if you're in the nervous system, you end up developing symptoms similar to neurocysticercosis. All right, so if it gets into the brain, you're going to start exhibiting things similar to neurocysticercosis. These things could also metastasize and go inside the bone marrow, where these are much more dangerous than, let's say, tinea would be if it got in there, because these things can enlarge, and as they enlarge, they start blocking the marrow, start causing necrosis of the marrow, and ultimately causing a thinning of the bone. That's typically a breakage of the bone. Is typically what alerts people to, hey, you actually have a hydatid cyst in that bone. In unrestricted locations, like let's say in the body cavity, right, just in the mesenteries, you can develop very, very large hydatid cysts. Size of a bowling ball, say. Some of the largest have been up to 15 quarts of liquid. And that they have millions of protoscolices inside of them. Now, just imagine 15 quarts. How many gallons is that?
3 3/4.
Yep, 3 3/4 gallons. How much does that weigh?
A lot.
Ballpark, right? If we say 8 pounds, 8 pounds per gallon of water, a little more than 8 pounds a gallon, but it's a good approximation. 8 pounds a gallon per water. We are 3 3/4. We're dealing with what, 30 pounds of fluid, 30 pounds of extra weight just in your body cavity, bulging.
So I would understand like someone not getting treatment if they have like say no access to like healthcare, but how could it get that bad to someone who like does have access to healthcare, like getting that large?
Any different reasons? I mean, I think I had made a joke when my wife was pregnant that she had a hydatid cyst. She was not amused. She was not amused. But you do have to think psychological aspect of some people. They start saying they know something's not quite right, but then they go in denial. They say, "Oh no, I'm doing fine. I'm healthy. You know, blood pressure's good. Everything else." Just afraid of what it is, and really, it's just the excess weight. Maybe they've got a mass, and they go in, and person's like, "Well, it's old age or something like that," or they mistake it for something else. I don't know. I think in most of these cases, they're individuals that don't really have the healthcare access. Now, other than the excess weight that the individuals are carrying, and you can imagine extra 30 pounds trying to escape. In this case, in like a human case, you could say escape some sort of large feline predator. All right, because again, we could be in intermedios, we get eaten, transmit it, but that extra 30 pounds slows you down. It's going to increase your predation rate if we are the prey item. But the other thing is this: the risk of rupture. Now, granted, if it's granulosis, it's got a thick acellular layer. It's going to be a lot harder to rupture. But if it does rupture, we're talking about almost near instantaneous death. And it's from anaphylactic shock. It happens so quickly because so much antigen enters your body just all of a sudden ramps up, and within a minute, you're probably going to be unconscious. And you're not coming back from that. Now, if we have alveolar Echincococcus, that is specific to, so this is a lot about the granulosis. Could also apply to alveolar stage. So if you get pinching it into the bone marrow and nervous system, that could definitely happen. This is definitely your granulosis, your unilocular hydatid cyst. The alveolar Echincococcus is all about getting infected with multilocularis. If you happen to get it, usually the first symptoms tend to be this pain or discomfort in the upper abdominal region. You develop some weakness, develop some weight loss. Primary site of these guys is the liver, and that's really where they're at, where they're spreading. All right, and because of that, then you can mimic liver cancer or cirrhosis of the liver. So you go into a doctor that doesn't have the time, or maybe if you don't have insurance and that sort of thing, doctor could kind of pass it off and say, look, you might have either liver cancer, or hey, this is, you're later in life, maybe you had one too many beers. This could be cirrhosis of the liver, and kind of leave it at that, even though this is a treatable disease. It requires surgery. So, I apologize to some. This is on YouTube. Tell you what, I'm going to hide this. This is a surgery that they are removing. There's one. There's more. And this is one individual. All of those, possibly only a single egg gave rise to all of this. Oh, there's one. You can see the fluid-filled sac. Fluid-filled sac.
I'm probably gonna regret asking this. But other than the egg, like, what's the yellow substance that's coming out?
That, I believe that's tissue fluid, host immune responses. So you could say lymph fluid, probably. Plasma. Yeah, we're not done yet. There's another bedpan. I like it listed as a live leak on that. You see that? It's not a live leak. This is just a one. The official one is actually Medical College, I believe, where they did this. So it's definitely that's the liver. I mean, geez, see the space that is just kind of hollowed out, and all the pressure, it can cause necrosis, so death of the liver tissue. Yeah, they're irrigating it to kind of get more out. Here's some. Just look at the size of that one. So I mean, they're squeezing it out, doing their best not to cause these things to pop. And the official one actually puts the bedpans together of all of the ones at the end that they removed. And they took 3 bedpans. That's how much they recovered. So that 10, 15 pounds extra weight had to be. So pretty nasty. And that's it. I know one of the front, they had it. This one. So, I mean, just those are all individual cysts. Now, in the human host, those probably don't have protoscolices. A lot of the ones that pinch off don't. So, Echincococcus. Any questions? All right. Moving on. Hymenolepis. So it's in the family Hymenolepidae. It's a large family. Members infect birds and mammals. The one that we're interested in is Hymenolepis, because we had a slide of that. And we did have a presentation. It's up. It's up online. You could hear it. So kind of get a second talk. That was up. Those were assigned before I knew we were talking about it, or I ultimately decided, hey, let's just talk about it because we do have a slide. And this life cycle is pretty darn interesting. So 2 species that infect humans is Hymenolepis nana and Hymenolepis diminuta. It's notable that all of them require an arthropod intermediate host. All members of this family except Hymenolepis nana, which is the one we're going to focus on. Now, we've seen our proglottids in lab. Wednesdays we'll look at them today. And hopefully you remember the testes were numerous and all of the proglottids that we have, except some of the ones that we've prepared in the lab upstairs, in the research lab. Hymenolepis actually has 3 testes in them. 3 testes, so you can see it. So in this group, you're going to have anywhere from 2 to 4 testes. And that's different from all the other ones that we've seen. The genital pores are unilateral, meaning they're all going to be on one side. And you do have a large external seminal vesicle. So seminal vesicle will be noticeable. The slide that we have is of a cysticercoid stage, different larval stage set, different from cysticercus. So cysticercus larvae, that's in Taenia, cysticercoids are in Hymenolepis. All right. So Hymenolepis nana is called the dwarf tapeworm. This is probably one of the most common cestodes of humans. It's estimated that about 1% of individuals in the southern U.S. are infected with this tapeworm, which you would say, well, that's not too high, but when you consider that the vast majority of people aren't infected at all, it's kind of high. The side that we have is a cysticercoid, a larval stage. So our cysticercoid consists of a invaginated scolex at the anterior end, and then a tail, which has basically been described as the cercomere that has been retained. Or a cercomere that kind of would be equivalent to the cercomere of your procercoids. So let's go over our life cycle. Leave the life cycle up on the side. So this is the life cycle for Hymenolepis nana. Even though I said Hymenolepis diminuta could also go in humans, we're not going to diagram that life cycle. This is just Hymenolepis nana. All right, so our definitive host will include humans, mice, and rats. And you could then kind of piece things together. Hey, they tend to live together, right? Overlap, association between all of us, so definite potential to get infected. Our adults are in the small intestine. Adults are in the small intestine, where the proglottids will get released. So, apolysis. They will rupture, break apart on their way out, so that it's the eggs that get released in the feces. If the proglottid hasn't completely dissolved, if it hasn't been completely emptied, some of that proglottid could come out and the eggs still get out. And the eggs are going to get consumed by our intermediate host, which is a beetle. Several different beetles. This is a popular one to study because you can use grain beetles or flower beetles, and you could raise it in the lab. Have cysticercoids go into these beetles. It just takes a glass jar. You raise them there and then feed them to a colony of mice and get them infected. So inside the beetle, and the beetles, both adults and larvae. All right, so larval stages or the adults can have. Now inside the beetle, you're going to have your oncosphere. That's in the gut, penetrates out, develops into our cysticercoid. Cysticercoid larvae, that is in the hemocele of our insect, our arthropod host. We then consume the beetle with that cysticercoid, and our cysticercoid, the scolex evaginates in the small intestine, where it then attaches and develops to our adult. Really interesting life cycle, right? Almost? Not quite yet. So, this is a simple life cycle. However, that's our human, mice, and rat host. This is our host. We can also consume the eggs. And if we consume the eggs, we now have our oncosphere that is in the gut. So the question is, where do these things develop? They have to develop to a cysticercoid. And then the cysticercoid is what develops into the adult. So where does it happen? Well, our oncosphere hatches, the egg hatches, releasing the oncosphere, and then what these things do is penetrate an intestinal villi. intestinal villi. Once it gets in the intestinal villi, then we have development to our cysticercoid. I'll just abbreviate intestine, villi. Now, once it gets to the cysticercoid stage, now it's ready to develop into our adult. So then you will have release from that villi attached to the small intestine. So release, scolex vaginates, attaches to our intestine and develop to the adult. The reason this can work is because our cysticercoid in this species can develop at a higher temperature. So temperature is one of those cues that kind of prevents things or allows the parasite to know which host it's in. We mentioned it for a couple life cycles already, where it's that increase in temperature was a signal, hey, we need to start developing, or we might be in the correct host. The reason this can happen is that cysticercoids can develop at that higher temperature, body temperature of our vertebrate host. So this is a way in which, you think about it, our host is kind of serving as an intermediate host in the function of producing the cysticercoid, and as a definitive host in terms of producing our adult. Now, I wouldn't call humans, mice, and rats necessarily a true intermediate host, but that's kind of how they're functioning. Because if you have the cysticercoid in there, and then our mouse or rat gets consumed by a human, on a desert island or something like that, who doesn't cook his food, yeah, you can then get that adult in your small intestine. So, are we done?
Based on that question, no.
Nope. Also have this. We have autoinfection. And that is, that egg hatches. So the proglotta gets released. It starts breaking up, releasing the eggs in the intestine. And you have the possibility then that the eggs actually hatch while you're in the intestine, releasing the oncosphere, which then finds its way to the intestinal villi, penetrates into it, and developing into a cysticercoid. We haven't seen an autoinfection like this at all. All of our parasites have required some way to move from host to host, except this one. This one can do it. You shed eggs out into the environment, eggs get consumed by a beetle, or they get consumed by another vertebrate. So, if I'm infected and I don't wash my hands and start cooking meals, there's a possibility I could spread those eggs to my family. All right, that's one route. Or I don't wash my hands and I go to bake some cake or bread or something like that, and I contaminate the flour that flour beetles get. All right, you have that route. Or once I get infected, my population of these worms inside the gut increases, not because I'm consuming more of these guys or more of these guys, but because the worm's replicating. The eggs are hatching before they get out, and now I have this autoinfection. So it has the potential to produce pretty heavy infections in an individual. Questions? In terms of adaptations to increase transmission, this is one. Or we can think about it at this point, because now we don't have to rely on this second host. And we don't even necessarily need to rely on the eggs getting out, but if we increase our population through autoinfection, now we're increasing the number of eggs that get out in the environment, which then increases its transmission to a different host. All right, so intermediate host is optional in this life cycle. We don't necessarily see it. So pathology, very limited, except in these heavy infections. And those heavy infections typically develop through autoinfection. And if you get it, then your symptoms are going to be similar to any of the adult cestodes. And I referenced Taenia saginata because we don't, again, this parasite isn't one that shows that pernicious anemia. We don't see that, but we see some of the other symptoms. Especially with heavy infection, you're probably likely to see loss of appetite. Probably some weight loss associated with it. Questions? All right. Our last family, Dipylidium. This family is similar in traits as Hymenolepis. The exception being that they usually have more than 12 testes. And we do have one that in our lab, it's called Dipylidium caninum, which is the dog tapeworm. Why do you think it's called the dog tapeworm? Found in dogs. Found in dogs. All right, so this dog tapeworm, we have it in the lab. I feel like the life cycle is interesting, but also the morphology is fairly interesting. Say it's interesting because we have two sets of reproductive structures in each proglottid. So we've got a separate set, male system, separate female system. And in order to accomplish it, we've put a genital pore on both sides of the proglottid. So you'll see this. We have this parasite. You have this parasite. You can check it out. Now, I think I have on the word list, thank you, Kyle, that I had uterus to identify. The uterus disappears early in development. So it's gone. You can make out the uterus in our Taenia 4 segment, Taenia slide. You can actually see the uterus without any eggs. But on this guy, we can't, because what ends up happening is the uterus starts disappearing in development, and then your eggs form in these structures, which are basically packets or clusters. So it's not, you have individual eggs, but they are in packs of maybe 5 or 6 eggs together in what's called an egg packet. So when the eggs get released, or when they get released from our proglottid, they're not individual eggs, they actually get released in these packets themselves, in these clusters. All right, so let's go through our life cycle. Got it. This is the life cycle of Dipylidium caninum. Dipylidium caninum. And you're right, it goes into dogs, but also cats. Dogs and cats. And inside these, we will find the adult in the small intestine. That's probably a good guess. If you're not sure where the adult is, small intestine, pretty good. All right, so how do the eggs get released? It's actually the gravid proglottids that get released. So it's apolysis. Apolysis. And proglottids are what comes out of the host. Now, they come out in the feces, or they actually wiggle out. Because again, proglottids have musculature in there. These things can actually wiggle out of the anus without having feces. And if that happens, then these things are around the perianal region. And that's typically where they need to go. Now, these things are going to look kind of like cucumber seeds. And you'll know it if you have a dog or a cat. You'll see these. I think some people call them grains of rice. When they dry out, yeah, they look something like that. But if they're fresh, they come out there, they're on the perianal region, and then once they get out, they dry out. So desiccation. And that releases then our egg packets. Our egg packets with the eggs. These things get consumed by our host. All right. So our host in this case are fleas. All right. But adult fleas feed on human blood. Larval fleas feed on organic material. So in this life cycle, it actually goes into larval fleas first. Fleas is our intermediate host. All right, so it's the larval fleas that feed on this organic material where you have your oncosphere that's in the gut, bores out of the gut into the hemocoel where it forms a cysticercoid. So same type of shape. And it's in our hemocoel. And I forgot to say, on our figure, we had the cysticercoid for Hymenolepis. I had a figure of that cysticercoid in the intestinal villi. I'm going to show you that again. I meant to say it. Right here, this is a cysticercoid that develops. The cysticercoid that develops in these intestinal villi are actually, they look different. They're morphologically different. Or you can distinguish between those that developed in a villi and those that developed in our beetle host. So the slides that we have look like the ones that are in the beetle host. They've got a much longer tail tied to it. But in general, even for this Dipylidium, the cysticercoids all look fairly similar to each other. All right, so we have a cysticercoid in the hemocoel. All right. And then I'm just going to do a dashed line here because we have metamorphosis of our host. Metamorphosis. So now we have our adult flea with our cysticercoid in the hemocoel. So our parasite actually survived metamorphosis of the host. The adult fleas didn't pick it up. They retained it during our metamorphosis. That's why I said our intermediate host is a flea. Why is that? Because I think this is an adaptation to increase transmission. All right? These guys, these larval fleas, aren't going to really stimulate any sort of response from our dog or cat. They're feeding on organic tissue. They're not feeding on the blood. But if an adult flea bites you, it's going to bother the dog and the cat. You're going to see them scratch. You're going to see them try to bite to get it. When they do that, they accidentally consume that adult flea that has a cysticercoid in it. Cysticercoid then, Scolex vaginates, attaches to the small intestine, and we have development to the adult. So this isn't quite auto-infection. So even though the host can kind of get magnification, egg gets released, goes into another host, and then you get a reinfection event. But a reinfection event is going to be much more likely with the parasite like this because that host, the intermediate host, actually live in association with our definitive host. Questions? Yep.
Does it affect anything about the metamorphosis? Does it speed it up or slow it down?
Not that I know of. I don't think we've seen it. I don't think we've seen it. Now, this can go into humans. And I had a former student whose sister got it. They will remain nameless because they felt extremely embarrassed. All right? Majority of the human cases are children. Can see some adults. So the question is why? Why is that? Is it because the adults are more resistant? Or is it possibility that the children are more likely to consume fleas? Now, I'm sure, how many of you have a dog or a cat? Some of you. How many of you actually have grabbed them and like let them kiss you and you like rub your face in them, right? And, oh yeah, you've done that. If they have fleas, you accidentally ingest one, possibility that you could get Dipylidium if they have it. So I kind of think maybe children are more likely to do that, more likely to let the dog lick their face over and over and over again. And see our stupid dog do that with my daughter. Let him get the worm. Let him get the worm. Actually, no. Our dog hasn't had it. It didn't get him. Because if it did, you can bet we'll have some live specimens in the lab. You'll see it. You'll see it. When we grew up with cats, if the cats go outside where they can get the fleas, there is a chance that you'll bring this in. It's pretty harmless to them. In terms of cleaning the litter box, you'll see the proglottids in their feces. You look at their back end, you'll see the little whitish stuff around in the perianal region. You'll see it with some dogs. You'll probably get alerted to it first, possibly, but they're itching or scratching. By that time, if they're itching or scratching, you probably have a cycle that's happening on the dog itself. It's nothing necessarily that you did, because if you take your pet outside, they can pick up the fleas just from nature. Dog parks, definitely dog parks, make sure you deworm them and so forth. And a lot of the dewormers will take care of the tapeworm. If we get it, we're going to have minimal pathology. All right? Again, kind of depends on your load and Tinea saginata, because we're dealing with the adult tapeworm. I think most people are alerted to it because they use the restroom and then they think, well, I didn't have any cucumbers. And then they put two and two together, and then they're like, okay, something's happening. And if you see it moving, good chance that it's a proglottid. All right, questions? All right, so we wrapped up the cestodes. Now we had one more. We have eggs of Menesia expansa. I chose to cut that out. We're not going to have, and we're taking that off our word list, so we don't have to look at those eggs. Because I quite frankly didn't know if we would get there. And definitely, I was still planning on to do the test on Friday. But I think with the number of life cycles that we've just covered, all right, and the information, I do think it's better if we delay the exam. So, we could do the exam on Monday, but I think I said already Tuesday or Wednesday. So I think what the exam's going to happen, what I'll do is I'll make the exam go live on Tuesday morning. You'll have until the end of the day on Wednesday to do it. All right? That gives you time to like study. Not only that, so it's not going to be the only thing. We have our practical that'll go live Monday, and you'll have all week on to take it. All right, so on Friday, so that way you can get into the lab and do some reviewing before you take that. I will also put up 2 quizzes on these. I'll ask that you get those done by Sunday. Well, we'll do like the general one that doesn't really ask much about the life cycles. I'll have that due on Friday, and the life cycles due on Sunday. So that way, if everyone takes it, and that's going to be our hard limit. If you don't take it, we're going to close it. I'm just going to assign zeros so that the key goes open on Monday. All right, we'll lock that in. Yep.
So 2 questions. For the practical, will it just be like identifying structures just like the last?
Yeah, identifying structures or the function. I might say, what is the function of these?
And then since the exam is going to be open Tuesday and Wednesday,
Yep.
are we still going to have class on
Yeah,
Wednesday?
we'll have class. So take your time, review the notes. If we have questions, we could take some questions on Monday. Or I'm sorry, take some questions on Friday. We can also take some questions on Monday. And I'm sure that's when you'll get bulk of your studying in over the weekend. So if you have questions about, if you don't quite understand something, feel free to ask at that time. And I'll have a better idea of how long it is at that point. I think the practical will likely, I'm going to target like 50 questions, 50 answers. So that's my target for that.
And how much time do you have on the test for the 50 questions?
For the practical, for the 50 questions will be like hour and 15 minutes maybe. About an hour, so it's like I have to see, it's between a minute and 15 and a minute 30 per question. And I do that because it'll be like one question kind of at a time. You can go back, but just it's harder to open it up and video it. I've seen that happen.
Wait, open up the quiz and people video the questions?
Yeah, and then you video the questions, and that way you have a copy of it. It's a lot harder to do it if you have to click through everything at the same time as trying to take your own in that own quiz at that allotment. It's a lot harder. See, I do have a lot of work to do. Yep.
How much material is going to be from the student presentations in lab?
Student presentations, it'll be estimate 1 question per presentation. All right, and we're looking at, of course, the Hymenolepis. We've got that covered. That was part of our slide. We'll change that in future years. But the other stuff, look for like the interesting information, the strange information. So we had one at Echincococcus. Goes in sharks, skates, and rays, and that's it. That's those. I mean, you can say, well, those are a lot of hosts. Yeah, it is, but also when you realize how many species are in that genus, I mean, it's quite impressive. And we've had. I'm trying to remember what other ones. Eubolifer. I mean, black spot. I mean, that's its name. So it'll be things that it's not going to be like, what is it? Unless the host is something unusual, it won't be like super specifics. Yep.
About how many questions do you think the exam will have, and then how long will it be?
Probably about 60 questions, maybe. The plan is to make that no more than an hour and a half. Hour to an hour and a half. But again, with the multiple choice, I think the rule is high school, you give them a minute and a half. And then I think for college, it's supposed to be a minute per question. But I think anatomy does 1:15, a minute 15 per question. And that's kind of what I've been targeting. Although that's like a true multiple choice. That's why I said like some of my multiple answers. I think 1.5 is probably a little bit more. All right. Next up, we're going to be moving to the nematoda. Fortunately, I didn't finish my introduction. I thought I was going to be good until next week. But we're going to move to a new phylum. So the second unit is nematodes and acanthocephalans and probably pentostomes as well. It'll be a much shorter period between this exam and the nematode and acanthocephalan and pentostome exam. We'll only have a couple weeks in lab, maybe 2 weeks. That's no big deal because we don't really have a huge number of specimens to look at for the nematodes. I kind of say once you've seen one nematode, you've seen them all. But it's not necessarily true. All right. See, some of you